Hi everyone. Today we are still learning about cytoskeleton since we already learned about intermediate filament. Today's lecture we are going to learn about microtubules. So microtubules have a crucial organizing role in all eukaryotic cells. Microtubules are long, relatively stiff, hollow tubules of proteins. They can rapidly disassemble in one location and reassemble in another location. So they are very important part of our cytoskeleton. So in a typical cell, these microtubules can grow out from a small structure near the center of the cell called centromere, and they extend towards the cell periphery. They create a system of tracks within the cell that these tracks act as highways for these cellular components and cellular vesicles to be transported to different between organelles and to different locations in the cell. So microtubules therefore are one part of the cytoskeleton. They are mainly responsible for transporting and positioning the membrane enclosed organelles. When a cell enters to the mitosis, the cytoplasmic microtubules disassemble and then reassemble into the mitotic spindle. So microtubules can also form cilia and flagella. So let's first look at the structure of microtubules. So microtubules are actually built from these tubulin subunits. So each tubulin subunit is a dimer with two globular proteins in the subunits. So each of these subunits are called alpha tubulin and beta tubulin. These are actually bound together by non-covalent interactions. So they are actually forming the wall of hollow cylindrical microtubules. So when these tubulin dimers stack together and bounded non-covalently, they will form a microtubule. So this tubule-like structure is actually made out of 13 parallel protofilaments. So protofilaments are the ones that when these tubulin subunits bound to each other, they will create a protofilament. When 13 of these protofilaments come and arrange themselves in parallel manner, it will create this hollow cylindrical structure, which is the microtubule. So each protofilament has a structural polarity because one side it will have alpha tubulin exposed, the other side it will have beta tubulin exposed. So therefore, each of these protofilaments will have a structural polarity. So the alpha tubulin end is called the minus end, and beta tubulin end is called the positive end of it. So here is what I was trying to explain. So microtubules are made out of these tubulin subunits called tubulin dimer. So each dimer has alpha and beta subunits. So these are non-covalently attached to each other like this to make a protofilament. So therefore, each protofilament has polarity, structural polarity. In one end, it will have beta subunit, and then the other end, it will have the alpha subunit. The one end that has the alpha tubulin is called the minus end, and the other end that has the beta tubulin is called the positive end. So 13 of these protofilaments can come together to make a hollow structure, tubulin, microtubulin protofilaments. 13 of them here, as you can see, came together to arrange into a cylindrical structure, which is the microtubule structure. So they also, you can see the electron microscopic structure, picture of a microtubule, cylindrical structure, and a cross section which shows that 13 protofilaments in the cross section. So another important thing when we come to the microtubule, like one of their major function is arranging the organelles as well as involving in cell division. So inside cells, these microtubules grow from specialized organizing centers called centromeres. So centromeres are typically can be found near the cell nucleus when cell is not in mitosis. So centromeres actually responsible in organizing these microtubules that radiates outwards through the cytoplasm. So centromeres actually, you might have remember that they contain pair of centrioles surrounded by a matrix protein. So this centrosome matrix contain a ring-shaped structure formed from lambda tubulin. So each lambda tubulin ring is the starting point for growth of one microtubule. So each centriole arranged in a perpendicular to each other and made of cylindrical array of short microtubules. So here is a picture of the pair of centrioles surrounded by this lambda tubulin ring. So they have these rings in the centrosome matrix, and each of these rings are a starting point for a microtubule. So microtubule will negative end will be embedded in each of these lambda tubulin rings, and they will continue to grow from the positive end, and then it will go extend towards the other end of the cell. So that is involving in the cell division, where as you know, during the chromosome segregation, these centrioles, these microtubules attach to the DNA itself and pull them towards the each end of the cell. So another function of microtubules is their involvement in cellular transportation, intracellular transportation. So in living cells, it is observed that under the light microscope that cytoplasm is in continuous motion. So mitochondria and small membrane enclosed organelles and vesicles seems to be traveling in a small jerky movements. So these movements are different compared to a Brownian movement, which is random thermal motions. So these solitary movements can occur along microtubules or actin filaments. And these movements are driven by these proteins called motor proteins. So motor proteins use energy derived from repeated cycles of ATP hydrolysis to travel steadily along these microtubules. So motor proteins can be divided into two families. So those are, one family is kinesins. They are generally moving towards the plus end of a microtubule. And then we also have another family which called dynein. These motor family, they generally move towards the minus end of the microtubule. So microtubules therefore involve in intracellular transportation. So a lot of the time, organelles and certain vesicles can move along the tracks of microtubules. So motor proteins are using energy to transport these vesicles and different cargos. So two families, like I mentioned, kinesians and dyneins, they depend on the polarity of the microtubule. So kinesians move towards the plus end, while dyneins are actually moving towards the minus end. So let's look at the picture. So as you can see here, you can see both the dyneins and kinesins. So those are motor proteins that will utilize ATP hydrolysis to transport certain cargo molecules along the tracks of microtubule. So their directionality of the movement will depend on the plus or the minus end of the microtubule. So when you deeper look into these kinesians and dyneins, they are dimers that have two globular ATP binding heads and a single tail. So these globular heads are enzymes with the ATP hydrolyzing activity, so they can hydrolyze ATP. So motor protein will bind to microtubule in only one direction, um, which is the head direction, and the tail will bind to a cellular component or their cargo protein, or some anything that they want to carry in between organelles. It could be a vesicle, it could be any cellular structure or anything like that. So the globular head, which has the enzymatic activity, can hydrolyze ATP, which provides the energy to drive this directional movement along the tracks of microtubules. So it goes in several steps. So when they move along, it's first the cycle of binding, releasing, and rebinding of that head part to the microtubule will happen. So the ATP will provide energy to actually move along this microtubule to each of these motor proteins. Another main function of microtubules is involving in cilia and flagella, which is actually a cellular movement. So many microtubules associated with other proteins and become stable to prevent dissociation. So cells actually take that advantage to use these stable microtubules to build stiff support such as cilia and flagella. So these microtubules actually, when they arrange into cilia, they are cilia actually the hair-like structures about 0.25 micrometers in diameter, which they cover the plasma membrane. So they extend from surfaces of many kind of eukaryotic cells such as respiratory lining, and their movement is very important to remove the mucus out of our respiratory tract. So sometimes cells use these microtubules in flagella. So when they use it in the flagella, they actually involve in movement of those cells, like swimming-like motions. So each cilium contains a stable core of microtubules arranged in a bundle that grow from the cytoplasmic basal body. So the motile cilia actually can beat in a whip-like fashion. So these movement help cilia to move stuff over the cell or give them the locomotion ability to single cell. Especially certain cells such as protozoa, they use cilia to collect food and also for their locomotion. Cells have cilia in the epithelial lining of the respiratory tract that allow them to remove the mucus and the debris that actually collect in our respiratory tract. So both the cilia and flagella are very important. So as you can see now, they use these stable microtubules to create these cilia and flagella, which actually use that stable structure to beat. So when they beat, they can allow cells to transport themselves, swimming motion. Also, they help cells to move particles out of the cells, like in the respiratory tract, or for protozoa to collect food particles. So here is the basal body and the structure of a cilia's power stalk. So they can move in a whip-like fashion when they are actually working. So flagella, on the other hand, which is also very important cellular structure for organisms, especially microorganisms, they use the flagella to move around. So if you compare flagella with cilia, flagella are much longer than cilia, and their major function is to move the entire cell. So flagella can propagate entire cell through the regular wave along the length. So both cilia and flagella have similar internal structure, where the 9 microtubules come together arranged in a ring centered with the pair of single microtubules. So this is called 9+2 arrangement, which is characteristic of almost all eukaryotic cilia and flagella. So here is that 9+2 arrangement of microtubules in cilia and flagella. So cells utilize that stable structures of cilia and microtubules to use these cilia and flagella for motion, locomotion, and removal of particles from respiratory tracts and stuff like that. So in summary, we learn about microtubules, which are stiff halotubes formed by globular tubulin dimers. So you now know that tubulin dimers contain both the alpha and beta tubulin. And then tubulin dimers can come together bound by non-covalent interaction to make these tubulin protofilaments. And these tubulin protofilaments, when the 13 of them come together, they will make a hollow cylindrical microtubule. So microtubules' main functions involve organizing organelles, cellular organelles in their respective places, and they also involve in cell division. These microtubules go out from the organizing centers such as centrosome, um, and they are actually embedded from the minus end and they grow from the plus end. And many microtubules actually can display dynamic instability, which means that they can uh, grow or assemble and disassemble from both ends, which is also important factor about microtubules. And then we talked about microtubules and their involvement in intracellular transportation. So two motor proteins involved in intracellular transportation are kinasians and dyneins. They associate with microtubules to transport cellular contents such as vesicles and organelles to different places. And their movement depend on the polarity of the microtubule. And then finally, we talked about eukaryotic cilia and flagella and how they use that stable microtubules to move whip-like fashion in the case of cilia. In flagella, they will use the stable microtubules to propel the whole cell in the media. So that is all about microtubules. I hope you enjoy this lecture. See you guys. Thank you very much.